So today we have two speakers. We have Nelson Clark and Miriam Demir. And Nelson will go first. Nelson joined the program in 2003 and had received a BS in Biomedical Engineering from the University of Miami before coming here. He's been working in Dr. Liu's lab in psychiatry, and he's been interested in looking at brain function using fMRI in various paradigms. And today, he'll be speaking about nicotine effects in brain and his publications. He's got a few of those, and he's presenting his work at meetings, and I believe he intends to finish this next semester. So without further, we'll just move on. So here, we'll listen to Nelson. Do you need a pointer?
Oh, no.
Okay.
Okay, good morning, everybody. Today, I'm going to talk about a project that we started out with as a collaboration with a group from China. My mentor, Dr. Liu, is from China and does a lot of collaboration work with them, the university that he got his PhD from. And the title of my talk is Investigating the Neurocorrelates of Smoke-Craving in the Brain using Functional Magnetic Resonance Imaging. The project overall had two major aims. The first aim was to investigate the smoker's perceived craving level and emotions induced by a three-tiered gradient of visual cues. And I'm going to explain what that three-tiered gradient is shortly. And the second major aim, which is something that I haven't been able to complete yet in this project, which is the more difficult part, and that is to evaluate the emotion of disgust in terms of its role as a deterrent in non-smokers and as a factor in determining the potential for relapse. Most non-smokers, such as myself, are perturbed or annoyed when you smell secondhand smoke or are exposed to it, and many in the field believe that that same sensation, whether you have it or not, is significant in terms of whether you're able to quit smoking or not, long term. Our first major hypothesis in this project was to use the results of the behavioral data from the evaluation of the stimuli, the visual cues that I'm going to describe shortly, and to correlate them to activation areas related to craving. Those particular areas are the insula, amygdala, basal ganglia, orbital frontal cortex, and DLPFC, which stands for dorsolateral prefrontal cortex. Another hypothesis that we have in this project is that there's going to be activation differences in the same conditions and the brain regions above within the different three subject groups. Those subject groups being a group of deprived smokers, satisfied smokers, and non-smokers. And we're hoping to see two separate three-tiered gradients. One three-tiered gradient is going to be between the low, middle, and high stimuli of visual cues. And another three-tiered and separate gradient is that we want to see people who are normally chain smokers but have been deprived of their nicotine have the greatest craving and the greatest intensity of arousal, with the people who are satisfied being in the middle group, and the people who are non-smokers at all in the third group. Let me briefly go over those three categories of subjects. In each category, we had only males, 15 males, and the average age was about 28. The first group was of deprived smokers, and they were prohibited from smoking for at least 10 hours prior to the experiment. The satisfied smoker was allowed to smoke as usual, including one cigarette right before scan. And both of these groups were chain smokers. The third group is a group of 15 males that had smoked less than 5 times in their entire life, rarely, if ever, picking up a cigarette. And I also forgot to mention there was a fourth group not included in this particular analysis, that will be used later in a further follow-up study, and that is a group of 15 males that were a successfully abstained smoker. This is a former addict who has been smoke-free for at least 3 months. Ultimately, the best clinical relevance that we would have for this project is to show similar neural activation patterns between the non-smoker and the successfully abstained smoker as compared to the satisfied smoker. The functional part, the functional MRI part of the scan consisted of 2 sessions with 117 volumes in each session and a TR of 2 seconds. So the total functional time was 117 times 2, which would be 234 seconds, which equates to a little bit less than 4 minutes. And then they were they were given a short break and then given a second set of stimuli. In the various blocks, the photographs were displayed on a screen for 3.7 seconds, followed by a 0.3 second interval, so that you had those photographs in those various trials been given for 4 seconds, which would include 2 TRs. We used a block design in which the group of photos were randomly selected from low, medium, and high related stimulation, as well as from a group of contrast process photos. Let me explain what I mean by the contrast process photos. And as you can see from the screen, this is an example of low stimuli. Some subjects just seeing a person holding chopsticks, which is basically a neutral stimuli. Although some people who are hungry, I guess, might get excited. And to the left, you have the same image, believe it or not, but in smashed, cropped up pixels. And the reason for doing this for all the photos was to have a control, because the highest amount of neural activation in any fMRI study is visual. So if we can account for the visual activation of color and the visual activation of the various pixels, we can separate and attempt to isolate the parts of the brain that are more focused on craving, more focused on emotion, and more related to what we specifically want to study. So in all the photos we had, not directly in front of, but sometimes before, sometimes after, they were randomly selected for all the subjects, but we definitely included 1 contrast photo per trial. And another example of low stimuli would just be somebody holding a pen. An example of a medium type of stimuli would be an ashtray with a bunch of cigarette butts in it. I myself, as a non-smoker, would feel a sense of disgust by looking at it, but somebody who is an addicted smoker might feel some type of craving by seeing that. The same is true for having a lighter. Another example of medium stimuli. An example of high stimuli would be a brand new pack of cigarettes ready to be opened, or seeing someone else lighting up a cigarette, who's sort of like seeing someone else, eating, your favorite dessert or something that you really want. That would be the highest amount of intensity for someone who's an addicted smoker. So before I get to a few of the imaging results that we have processed so far, let me just briefly go over some of the behavioral data that we have thus far, that we've thus far gathered in the project. And as I mentioned before, there's 2 separate 3-tiered gradients that we wanted to display and express. And one of those being the difference between the high, middle, and low or non-related stimuli. The non-related, unrelated being, the pen or the chopsticks. And as you can see based on this graph, with the lower scores indicating a higher craving level of self-reported surveys data that we collected from these subjects before scanning, not during scanning, but before scanning. You can see that from self-reports, for all 3 groups, they reported a higher level of intensity or craving for the more intense stimuli. whether they see a pack of cigarettes or they see somebody lighting up a cigarette, they're more inclined to say, yeah, that would induce more craving than just seeing an ashtray, which would be in the middle, or just holding a pen or chopsticks, which would be the least likely to induce craving. And in addition to that three-tier gradient of stimuli, we also see our three correlated lines of data within our three groups of subjects. The deprived smokers in navy blue were the 15 males who were chain smokers but were not allowed to smoke for at least 10 hours prior to scanning. And they almost are identical to the line in purple, which is the non-deprived smoker. I should have written satisfied smoker. The smokers who are also addicted but are allowed to smoke in general. And this makes some intuitive sense, because whether you're a deprived smoker or whether you're a satisfied smoker, you're both going to be showing that and have that same feeling of intensity. You're both going to be craving those cigarettes from those self-report behavioral data, even if you have been neglected for 10 hours without smoking. So we're not surprising to see those two lines very similar. And then, as we expect, the non-smoker to have the least amount of self-reported data, because those males, have smoked cigarettes for less than 5 times in their life, so they're not going to report a high intensity of craving for those stimuli. So as part of the behavioral data, we pretty much have gotten what we wanted. And the key to analyzing the imaging data is to be able to show that although there's a non-significant difference between the satisfied group and the deprived group in terms of behavioral data, in terms of what's going on in the brain, there are significant contrasts. And by showing that, we can say, well, although someone may say in a survey that he is not aroused, that he is not craving a cigarette, in reality what's going on in the brain is something different, which to a clinical person would be the most important aspect of the study, because then he or she can then figure out, okay, we need to try something else to get them off nicotine. These people are nicotine dependent. We need to figure out how to treat them differently. So in terms of approaching the data analysis, obviously we wanted group comparisons of those three subjects. We want to compare deprived versus non-smokers, deprived versus satisfied, and the non-smokers versus satisfied. And we also want to compare the three categories of stimuli, high versus medium, high versus low, and the medium versus low as well. And I'm not going to be able to mention this in my talk today, because to be honest, I haven't started on this part of the project, but ultimately we want to show how these subjects are reporting disgust versus craving, and model that in a separate two-dimensional plane. And that'll be something that we could also use as a follow-up for this study. So let me just briefly go over a little bit of the imaging data that we have processed so far. And I'm just going to go over the high stimuli versus the low stimuli in the three-dimensional brain. And as you can see, these three rotations are for the deprived group versus the satisfied group in terms of seeing lots of high pictures versus seeing lots of low pictures, and subtracting the two groups of activation. So we're trying to disregard visual processing, we're trying to disregard a lot of other variables that would go into the scanner, and just focus on the difference between those two types of stimuli.